Welcome back. Today we'll be going more in depth about the topic of leukocytes. So leukocytes, or white blood cells, compose 1% of all the blood, and in the normal range in adults it's about 4,500 to 11,000 per microliter. And of course they protect the body against invaders, and it also plays a role in repairing injuries. It's the first, it becomes functional in the tissue after leaving blood circulation, and they can be either granulocytes or agranulocytes, and they have something on them called P-selectin cell adhesion protein that binds to the glycosylated ligands on the leukocytes, and it slows down their transport. So the receptors for the adhesion proteins is going to pop up in, when the cell is distressed and in areas where there has been damage. So the P-selectin will adhere. The leukocytes are round, so they kind of tumble through the blood, and by tumbling, they adhere, and it slows down the transport, kind of like Velcro, and once the cell has adhered, it goes through diapedus, where the leukocytes will send its process between the gaps of the endothelial cells and then squeeze through the venules into the capillaries. And then chemotaxis is where the bacteria will secrete certain chemical signals that the leukocytes will recognize, and that's where they will go to find the bacteria through the diapedesis. Granulocytes are going to be terminally differentiated, and they have a lifespan of only a couple days. They have more than one lobe, and the cytoplasm can have a few organelles, a small Golgi, and a not very abundant rough endoplasmic reticulum or mitochondria. They really only rely on glycolysis for energy. They have the azurophilic granules that stain purple, so they're very acidic, and the specific granules will have specific functions, and they bind to different dyes based on the composition. So neutrophils will have they'll be pink, while the eosinophils will be binding to dyes and be stained orange pink. Basophils, of course, will be blue, and that kind of tells you the components of the granulocytes, or the granules. So, of course, like blue, it's going to be basophilic, so it's going to be the most acidic. Okay, so the granulocytes die by apoptosis, and then the debris is removed by the macrophages, and all of this doesn't elicit an inflammation reaction. Neutrophils are polymorphonuclear, so they have 2 to 5 extra lobes of their nucleus. It's the most abundant type of leukocyte, and in it stores glycogen for energy, though it relies on glycolysis for the main source of energy. The immature neutrophils make up a large minority of the neutrophils, and the mature neutrophils are the active ones. They survive in the anaerobic environments, and they're the first white blood cells to enter tissue during any kind of abnormality, so during inflammation or infection, and they move by an amoeboid motion, so it's they don't really roll anymore once they have to squeeze through using diapedesis, and they have to inchworm their way, crawl their way to the site of injury. In women, they have a drumstick appendage off of the nucleus, which is the bar body, so it's the inactivated sex chromosome. And the cytoplasm, of course, has very few organelles, and they have the azurophilic primary granules and the specific secondary granules. The azurophilic primary granules are going to be modified lysosomes, and they're usually in the young neutrophils. Those will fuse with the phagosomes to make a phagolysosome, and those major role is to kill and degrade engulfed microorganisms. and they use the enzymes to degrade the bacterial cell membrane, and then from there, more like bactericidal enzymes are like injected in. And we're using an NADPH oxidase respiratory burst, and then the hydrogen peroxide will eventually like lyse the bacterium. The specific secondary granules really only appear in the mature neutrophils, and they are involved in the inflammatory response to trying to like localize the infection. Chemokines are released to attract more white blood cells, and the cytokines are released to regulate the activity of those cells and begin the restoration of the affected tissue. So they circulate in the blood about for like 6 to 8 hours, and then migrate into the connective tissue and stay there for like 1 to 4 days before dying by apoptosis. And the apoptotic neutrophils, along with the cellular and bacterial debris, will form pus. EOSINOPHILs are bilobed, and they are pretty numerous. And they're large, and they have the specific eosinophilic cytoplasmic granules that stain like red to orange. And these really play a role in killing the helminthic worm, parasitic worms, and plays a role in lipid mediation. It also is an important part of regulating allergic reactions, and it will mediate the allergic reaction by inactivating leukotrienes and histamines. So they will phagocytose and remove antigen antibody complexes, and they are found in increased concentration during like reactions and the parasitic infections. So of course they'll be really abundant in the mucosal lining of the respiratory tract, the GI tract, and the genital tracts. Those will spend about like 4 to 5 hours in the blood, and then migrate into the tissue, and then survive for like 8 to 12 days. The cytoplasmic granules are kind of oval and flattened core, and those will contain the major basic proteins. So this is going to be a basic molecule, or basic cell. And the eosinophilic peroxidase, histaminase are the specific to these cells, and they kill worms by gathering around the worms and basically digesting it from the outside in. Basophils are rare. They're not really found in any significant amount, so if they're elevated, it's a pretty important sign that the patient is quite sick. This is an artist's rendition of it, but this is what they really look like under the microscope. And they are very strongly basophilic, and they are the circulating equivalent to the mast cells, and they have surface receptors for the immunoglobulin E, and those very basophilic cytoplasmic granules contain heparin, histamine, phospholipase A, which are all very acidic, and they migrate into the connective tissue, and then they can differentiate into the mast cells. And these are the ones that really play a role in like promoting allergic reactions. So the first exposure to an allergen will elicit the specific IgE antibodies, and then those will bind to the basophils and the mast cells, and then on the next exposure, the allergen will they will bind very fast, and then they combine with the IgE antibodies, coat the cell surface, and then trigger the release of the granules in the type 1 hypersensitivity reactions, and of course that will result in asthma, allergic rhinitis, conjunctivitis, and hives, and anaphylactic shock, of course, is the most serious. Agranulocytes are terminally differentiated cells. They can live for hours or to years, and they have the azurophilic granules, but no specific cytoplasmic granules. Monocytes are going to be the largest white blood cell. Their main role is like phagocytosis, and they circulate in the blood and then enter the connective tissue and then differentiate into the macrophages. They're also precursor to the Kupffer cells in the liver, the microglia, osteoclast, the dust cells, and the Langerhans cell in the pancreas. They're antigen presenting cells, so they play a role in the uptake, processing, and presentation of antigen to activate the other lymphocytes. And the cytoplasm is quite small, and they only have very little organelles. The nucleus is it's like C-shaped, or sometimes it's like a bean-shaped, and these respond to the presence of necrotic material and invading microorganism by increasing the macrophage population. So these are not particularly rare, but an increase is in serum is a bit of a concern. And in chronic inflammation, it's going to be high all the time. So platelets are thrombocytes, and they are small, non-nucleated biconvex disc. They have a lightly stained periphery hyalomere and a dark, intensely stained core, also known as the granulomere. The cell membrane is coated with a glycocalyx, and that will repulse adhesion to normal endothelium. So that's why our arteries and our veins don't clog up all the time. Instead, they will adhere to the injured vascular endothelial cell by binding to the collagen in the vascular basement membrane that is now exposed after an injury. And they appear in clumps, usually in a blood smear. They live around for 10 days, so don't be picking a scab before the 10 days, because they're still platelets doing their job, plugging up the hole. Normally it's about 150,000 or 400,000 per cubic millimeter of blood. Oh, sorry. Last one. Jumped the gun. Hyalomere is the marginal bundle. They don't really show it here, but it's going to be more towards the periphery of the cell, and it is really formed as the platelet is pinched off from a megakaryocyte. We'll cover that in the next lecture. And those really help maintain the shape of the platelet. There's going to be an open canicular system where the vesicles are made from the invagination of the plasma membrane that helps the uptake of growth factors and other proteins. The dense tubular system is going to be made out of irregular tubular vesicles that come from the remnants of the endoplasmic reticulum, and it stores calcium. And both systems really help the exocytosis of protein and when adhering to collagens and whatnot. Okay, so that's it for now. Thank you for your attention, and good luck studying.